I'd like to introduce Samantha Anderson. She is going to be a rising senior at McHenry West High School, which is in Chicago, Illinois. She's come up to Claflin to do her first research internship in molecular biology. She's been working in my lab for a week and a half, and so this is the culmination of a week and a half's worth of work. And I'll give it to Sam. Thank you.
All right. So basically, what Dr. Jessic has been working on since graduate school is redesigning beta-galactosidase so it can work at low temperatures. Basically, so beta-galactosidase is
Lower one.
all right. Beta-galactosidase, basically what it does is it helps lactose intolerant people, and it helps you. It's inside your body, and it cuts lactose into galactose and glucose in order to utilize the energy during glycolysis. And what lactose intolerant people do, they don't have that enzyme, or it doesn't work very efficiently. So lactose intolerance affects 30 to 15 million people in the United States, mostly among minorities of Americans, American Indians, and Asian Americans. And currently there is lactose-free milk or different types of milk that have beta-galactosidase in them in order to cut the lactose before your body can, because obviously lactose intolerant people can't do that themselves. So they have an enzyme that does it for them, but it only works in class pasteurization where it's really hot. And so basically it doesn't break down all the lactose in the milk. So we want to find a way for it to work in the fridge in order to break down more lactose, and so people can utilize the energy more efficiently. This is beta-galactosidase. It has beta strands and alpha helixes multiple times wrapped around, and we're trying to change different parts of the amino acid sequence in order to change parts of that so it works at a larger range, because, well, that's like this. And so we would need it to work at a larger range, so it works before they put it in the flash pasteurization, at the flash pasteurization, and when you move it into a fridge. So regular mesophilic, what you have in your body, mesophilic beta-galactosidase, works from 20 to 60. Not high enough for the flash pasteurization and not low enough for the fridge. So what we want to do is take a thermophilic beta-galactosidase and expand the range so it works at all three places. So what we know is, like, enzymes, their function is based upon their structure, and the structure of amino acids, and amino acids off your DNA. So we have to go back to the basics to change the entirety of it. And from previous research, Dr. Panasic has seen that you can expand the range, so we need to do more to expand it further. He's done a little bit. I think he got it down to 18 degrees Celsius, so we need to move it down to 4. And so then, in order to do this, you need to change parts of the DNA, but you can't change too much, because obviously then it'll change the entire function of the enzyme. But you can't change too little, because it may change a non-coding piece or something that wouldn't actually change any amino acids in the sequence. So in order to do it, you need to ligate an altered beta-galactose gene with plasmid, transport it into an E. coli, culture it, and observe, see if it works at lower temperatures than before. But first, what I'm here to do is we need to make a plasmid with just the beta-galactose gene in it with no changes, so you have an original to start from. So what was done before I got here was that Daniel used a Puck-18 plasmid, and we needed to get rid of the alpha fragment in it, so that you can tell later on, when using blue-white screening, whether or not the beta-galactose gene worked. So in order to do that, you take out the alpha gene, and we did that using an MD1 and HIN3 site. And what that does is it creates two sticky ends. The first one up there is HIN, and the next one is MD1. And using Clenau, we filled in the fragments so that you have these two pieces here. This is filled in from the HIN3 or from the ND1 site, and this is filled in from the HIN3 site. And this plasmid is what you have left. And so then what he did also was he purified with the PCR purification, ligated the two pieces together after they had blunt ends, transformed it into E. coli cells, plated it to find white colonies in the blue-white screening, and then he selected a colony, made a culture, and after testing with the gel, he wizard prepped the isolated DNA and transformed the DNA into 4 separate samples to test. So when I got here, we started to run gels, and this first gel is sample 1 and sample 2, sample 3, 4. And the first lane standard, second uncut, and then the third one, is P delta alpha H with PBU2, and the third one extra, and the next one ND1 and HIN3. So what was supposed to happen was this pattern here. You can't really see it, but if you look at this one, uncut runs here, then if it's cut once, it should be here. This one should be cut here, and then uncut and cut. These two did not work. You see the straight lines here. But this one, as you can't really see it, but it looks almost like this one, except this one here was totally digested, like this one only partially cut. So we decided to use transformer number 2. We isolated it, and then in ND1 site we put it in, so later on the beta-galactosidase gene would fit because it needs an ND1 site and a HIN3, or yes, a SAL site, but the SAL site is already in the P delta alpha H. So we used quick change PCR to insert ND1 site, and we checked it with a restriction digest, which is here. And here's standard uncut HIN1 or HIN3 ND1. Then we did a second one to test, and this one, the ND1 was supposed to cut after the change, and it did not, but here it did. So in sample 2, we decided to use again. And this is the new plasmid we had after we inserted the ND1 site and the HIN3 site. Alright, so then we have the beta-galactase gene, which is in a different plasmid, obviously, from the beginning. So PMA5 BJB, and then we put it into the E. coli cells, transformed it, plated, chose blue colonies this time because you're looking for an active BJB gene. Then you isolated the DNA, and now we need to insert a SAL1 site, because in this previous one, you have a SAL site right here. So they want to use SAL1 and NDE1, because this is, if you can see right here, this little area is where we want to insert the BJB gene, and so we wanted to insert a SAL1 site into the plasmid. So then we did a quick change PCR to insert a SAL site, and for the first set, we checked samples 1 and 2 just to see if they worked, and unfortunately they did not, because cutting with ND1 and SAL should have produced 2 fragments, and it did not in either one. So we returned to samples 3 through 6 to see if any of those worked, and basically none of them did. None of them both cut into 2 fragments for ND1 and SAL. So we had to start back from the beginning, and we decided to change, instead not use a SAL site, but we decided to use a BAMH1 site. So, we didn't have to do any PCR quick changes because it was already in, the BAMH1 site was already in the PMA5 EJB. So, and then how we isolated the DNA instead of doing the wizard prep or the PCR purification stuff, we used a flash gel, which was a new type of gel where you loaded the gels into one sample, and then you let it go, you let it run, but you can see the bands as they're moving, and then there's a second well, and you wait for the band to go into the second well, and then you just extract it with a pipette.